In the interest of time. Hey, everybody. Welcome on this super chilly Tuesday to CGIBD seminar. I'm really pleased to introduce today Dr. Nathan Crook, who's Assistant Professor of Chemical and Biomolecular Engineering at NC State University. Dr. Crook's training in chemical biology began with undergraduate work at Caltech, followed by doctoral studies at UT Austin. And then postdoctoral work at WashU in Gautam Danta's lab. Dr. Crook's lab is innovating ways to engineer biology, and looking at his website and his publication record, there's just a lot of exciting work going on in his lab with synthetic biology as sort of the central workhorse. I'm really excited for his talk today, which is entitled Engineering Probiotic Yeast for Controlled Synthesis of Drugs and Vitamins in the Gut. He's also a pilot and feasibility grant winner from the CGIBD, and I'm sure we're all excited to hear all about his work. So welcome and take it away.
Thank you. I appreciate it. I'm really honored to be here and give a talk to you all. It's been, I think that you'll see a lot of familiar faces in this talk. We collaborate with a lot of people, and so it's cool to be here. Before I get started, I have to say that I filed patent applications for a couple of the technologies that I'll talk about. Those are listed here, but the reason why we say this is because I have a vested interest in making them sound awesome. But please do put on your critical hat when you see that, and then ask me anything. Happy to discuss why they may not be awesome. I also want to thank the people who do the research. I'm of course not in the lab doing any pipetting. It's all these fine people doing it. Actually, the people who are in this talk, some of them have graduated, and I'll be sure to get their pictures on the sides of the slides that they're involved in. But we're really blessed to have a large group of hardworking people in my lab. So I'll begin by discussing a bit of a vision for what I think what drives me in this particular research. It's a bit of an out there vision, so please humor me, I guess. Our own body has tens of thousands of genes, many cells, and a large amount of biomass that it contains. So it can do chemistry at relatively high titers and a wide diversity of chemistry to support our life. But interestingly, and something that drives my interest, is the microbial community that we house within our guts. Compared to the amount of genes that our own genome encodes, it has orders of magnitude more, higher diversity of chemistry that can be performed in the microbes that exist in our gut, about the same number of bacterial and fungal cells as there are cells in our body, and 400 grams of biomass, which, if you've ever done a fermentation in a fermenter, is a non-insignificant amount of bacteria that can do high titer chemistry. So in its natural state, the microbiota, as I'm sure you all know, takes dietary fiber and converts it into molecules that can be used by our body, energy and vitamins in particular. But then the stuff that cannot be digested gets excreted as fecal material. Some of that is just summarized here, but in terms of quantitatively, 10% of our daily caloric intake comes from the action of our gut microbiota, and it also synthesizes K vitamins for us as well. Interestingly, from a sort of organ perspective, the microbiota is relatively easy to change. You can take a probiotic or a fecal microbiota transplant and change the chemistry that this microbial organ does. Additionally, the species in our gut, some of them are relatively easy to engineer. And so I'm not the only person to think about this, of course, but why not change our microbial bioreactor for good? Perhaps what you could do with this is to overcome illnesses that are associated with our microbiota. And then in the next two slides, I'll talk about human metabolism as something that could also be impacted. So as a chemical engineer, I've been a little bit, I would say, interested by this paradigm of how we do biomanufacturing for drug synthesis and drug treatment. So the way this works, and this is not, of course, new information, is that you're healthy and then you say you contract a disease, right? Maybe a microbe comes and causes an infection. You have to feel sick, right? You have to, sometime occurs and then you don't feel good anymore. And you have to see a doctor at that point, right? You say, okay, now I'm sick. Doctor, please help me. Your doctor tells you what to take. And hopefully, if you're lucky, a company has decided that it's profitable to manufacture the drug that you need. And that drug has been made somewhere else and it comes to your local pharmacy, stored there for a while. And then you buy it, it's still good. You take it and you sometime later you feel better. So this works really, really well. It works really, really well. It's sort of how we do things. It works particularly well in places that have money and have doctors and have places that make medicines. And in some cases, this may not be the case. And so for some diseases, some bugs, and some settings, you may find, I'm envisioning a situation like this, where you manufacture a probiotic, or maybe there's a food culture that is being sort of passed along over time that's stable, and you ingest that living microbe. There might be some money involved. You take that microbe, you contract a disease, but crucially, that microbe might be able to sense that you've contracted a disease and autonomously give you a therapeutic in response. Hopefully, so that you don't even notice that you were sick in the first place and you still feel healthy. This is kind of not a new idea by any means, but I thought I would describe the vision for what our lab would like to be able to contribute to. But knowing how to treat diseases that are obvious problems is only half the story, because human metabolism has some, I would say, bugs in it, some quirks that is you might want to change if you're thinking about it from a purely engineering perspective. For example, your body can synthesize its own glutamine, right? You don't need that in your diet. You can utilize other materials and synthesize that for yourself. Lysine, which is a related amino acid, cannot be made by your own body. You have to get that from your diet. So if you're unlucky enough not to have access to lysine, you are really out of luck. Same thing with carbon sources. Glucose is tasty, delicious, and carbon dense and calorie dense. Cellulose is also calorie dense, but your body doesn't know what to do about it. It cannot cleave those two sugar molecules and get energy from them. Vitamins, for example, are things that our body needs but cannot make. That's why they're called vitamins. So why do these things exist? Why can't we make our own vitamins? There are molecules also that are found in nature that are almost, I would say, reducing our ability to get nutrients from our diet. Phytic acid is a great example. It chelates iron. It's an iron chelating molecule that plants make to get their own iron. The unfortunate part is that if your diet contains a lot of phytic acid, the same amount of iron will not be absorbed by your body because it's competing with this phytic acid for that iron. And then, of course, there are the obvious antagonistic molecules that nature makes that you eat and your body does not metabolize, and in fact causes severe sickness if you ingest them. And so it would be nice if we could use the engineerability of our gut to address these issues. Perhaps if we could make our gut more designed for purpose, right? As you're growing, you could maximize the amount of calories you can get from any type of dietary source. Maybe people who are pregnant could have prenatal vitamins when otherwise they wouldn't have access to them. And finally, as we age, right, it's not about maximizing calories; it's about moderating the type of energy that we're able to intake. And so there are just a few examples of things that crazy people like me are thinking about doing with our gut microbes. But as with any crazy idea, there's the crazy idea, and then there's actually how we do it. Here's another picture that's equally vague and not perhaps as detailed, but it is going to be used to sort of organize this talk. And the talk has four main themes. And in order for us to really do anything to our gut in a safe and effective way, right, because we want it to be safe and effective, we have to be able to do several things, and four of them we'll talk about here. First of which is the ability to engineer the bacteria that we want to engineer, or the microbes. I'll be more general about that. The second one is the ability to sense the disease or sense the condition that we're intending to improve upon. The productivity of that chassis is something we would like to be able to tune, maybe keep it within a therapeutic window, or even better, modulate that to be in response to the extent of disease. Another thing that would be quite interesting to be able to do is to tune the abundance of that chassis, maybe for acute conditions there could be a treatment that only lasts a short amount of time. But for chronic conditions that happen in the gut, perhaps like cancer, for example, you might have a bacteria that establishes a longer term interaction with its host. And so I'll go through our efforts at sort of tackling each of these four problems. I'm not going to claim that I've solved any of them, but we're making progress, and hopefully that progress is as exciting to you as it is for us. So we've got with ability to engineer first, and the host that we're choosing for these studies is a bug called Saccharomyces boulardii. It's a probiotic yeast, and so yeast is a eukaryote. If you've done a lot of genome sequencing or metagenomic sequencing of the gut, you might be surprised because eukaryotes don't make up a large portion of the gut. By gene or nucleotide count, they're only about 0.1% of the genes that are present within our gut. However, eukaryotes have much larger cell bodies per nucleotide than bacteria do. In fact, yeast is about 10 times larger, 10 to 50 times larger than a bacterial cell is. So doing that sort of mass balance on nucleotides versus cell mass gives fungi a substantial portion of our gut space. And so they should be studied, and I think they represent promising chassis for delivery of drugs to the gut. So as support of this, here's a picture from a 2012 article where they stained the mucosa of mice for anti-yeast antibodies, and these yeast glow in red there. And so they're lining the mucus because they can't divide as fast as this material flows through. So hopefully they're eating that material there. Now, Boulardii in particular is a variant of baker's yeast, but it has the following interesting properties from a therapeutic perspective. It is generally regarded as safe. You can buy it over the counter and has a lot of antidiarrheal properties. But for us as engineers, it's genomically very similar to Cerevisiae. So Cerevisiae has been engineered for a very long time and has the ability to secrete designer molecules. And so, however, unlike Cerevisiae, Boulardii grows at body temperature and Cerevisiae grows at 30 degrees. So it's an interesting chassis to use for the gut. So the goal we started out was to develop engineering tools for Boulardii so that we could then deliver the biomolecules we wanted. So the first thing my student, Dennis, decided to do, she's now a postdoc at TTU, what she wanted to do is look at promoter activity in the gut. So she cloned these collection of promoters in front of GFP in yeast and measured the fluorescence output of GFP from Boulardii. And so you see that of these many promoters that she tested, we can get about a 50-fold range in gene expression just by changing the promoter sequence. Interestingly, these promoters were taken from Saccharomyces cerevisiae, but they still worked in Boulardii. Interestingly, ALD6 is a weak promoter in Cerevisiae, but a rather strong promoter in Boulardii. And so we're very interested that perhaps this difference in gene expression could be a result of its the different strains of yeast we were testing. With that promoter toolkit now, we wanted to try to produce something. And the thing we decided to produce, which is the vitamin portion of this talk, was beta carotene. So beta carotene is a molecule that looks like this. It's a precursor to vitamin A. If you take pure beta carotene in your diet, you need 12 equivalents of beta carotene for one equivalent of retinol, which is the active part or the pure vitamin A part. We were interested in this molecule for a couple of reasons. Number one, it was colored, right? So we knew if we were doing it right. Another reason is because vitamin A is important. Vitamin A deficiency is a devastating problem, and we're trying to perhaps enable the gut to produce its own vitamin A so that you wouldn't have to have as much from the gut. Another reason to be interested in vitamin A is that all transretinoic acid, which is a derivative of vitamin A, when you deliver it to the gut, has this anti-cancer abilities. And I think a lot of carotenoids in general have beneficial impacts to the colon. You see a lot of studies demonstrating their anti-inflammatory impacts in the colon. So we're interested in a way to deliver these directly in the gut. So my student, Scott, at the time, turned to another carotenoid-producing yeast called Xanthophyllomyces dendrochus. It produces beta-carotene natively via a very nice metabolic pathway shown here. When you compare its metabolic network to Boulardii's metabolic network, you need a few extra genes. Number one is an HMG1 enzyme that brings flux into the terpene synthesis pathway. CRIT-E takes metabolic flux from IPP and FPP, diverting it away from ergosterol and into the adrenal pyrophosphate. But then you have a few other genes, CRIT-YB and CRIT-I, that then convert that to beta-carotene. So it's a 4-gene pathway. When you're a metabolic engineer, the thing you want to know about is how high do I have to express these enzymes for our cell to not only grow, but then produce a lot of our desired molecule. You can imagine if I really ramped up the production of HMG1 or CRIT-E, I would increase flux toward my desired molecule, but it might do so at the expense of cell growth. The cell needs acetyl-CoA for other things, and ergosterol is a main component of its cell membrane. And so I don't want to deprive our yeast of those abilities. So what Scott decided to do is take that promoter library we had developed previously and choose 9 of those promoters in front of each of our genes. So CRIT-E is an example here that he did. He took 9 different promoters, CRIT-E and the terminator, and did an initial assembly of that cloning reaction. Now that generated an in vitro mix of all 9 plasmids, but he didn't transform that into yeast right away. He then did a PCR of that product, generating an amplicon library, 9 different amplicons per gene that we were interested in expressing. Now, the beauty of yeast is its ability to do homologous recombination. Now, with these 9 different amplicons per gene, we can mix them all together with our actual yeast backbone vector and co-deliver them into yeast at the same time. And the recombination machinery of yeast will assemble these 5 parts into an intact plasmid. It sounds really crazy, but yeast is really nice in its ability to do this. And Boulardi is no different. So Scott showed me this picture and I loved it. It was yeast that expressed different variants of this metabolic pathway. You see here, each of these colonies is a different color as what you get after your transformation. And the challenge now that Scott and Dennis had to do is comb through this plate to pick out the variants that look the most orange to them. And so they did this, and they got this collection of 28 different variants that we were then able to genotype and measure beta-carotene levels off of. You can imagine that some of those metabolites are colored that are not beta-carotene, so we had to actually use HPLC to figure out which ones were which. Okay, so now I have a strain of Boulardi that makes beta-carotene. The next thing we want to know is, does Boulardi live in the mouse gut? And so Ibrahim, and I actually should have Sue's picture here. Sue Tonkanaji helped us a lot with this study. But we did Boulardi in a couple different mouse models. One was a completely germ-free mouse, and one were 2 different types of antibiotic-treated mouse models. In the germ-free mice, what we saw was a stable colonization of Boulardi in the gut for up to the length of our experiment, which was 30 days. We could have gone longer, but we just decided, okay, we have to move on to other things. The other 2 treatments, the antibiotic-treated models, the blue treatment was a more intense antibiotic treatment, and the green treatment was more short-term. And so we see that as the antibiotic treatment is longer, you get a longer residence time of our yeast. So the take-home message of this is that the endogenous microbiota inhibits the colonization of Boulardi, and it does so in sort of a dose-dependent manner. For the purposes of our study, though, we said, okay, if our yeast can synthesize beta-carotene in the gut at all, we'll have the best chance of seeing it in a germ-free mouse. So that's what we went with. We did a second germ-free experiment, again, with the help of Dr. Tonkanaji, which we're very grateful for. And what we did is we took 2 groups of mice and gave one group of mice wild-type Boulardi, and another group of mice are beta-carotene producing Boulardi. In this graph, what you're seeing are the colonization levels of those 2 types of yeast. In the wild-type Boulardi, there were a little bit higher abundance than the beta-carotene producing Boulardi. But when you can look at the amount of beta-carotene present in the gut, the amount of beta-carotene is much, much higher in the beta-carotene producing group than in the non-beta-carotene producing group. There is still some residual beta-carotene in the wild-type arm because of the beta-carotene present in the mouse chow. But our yeast was able to live in the gut, synthesize that beta-carotene from available undigested material, and produce that beta-carotene, which we were very, very excited about. All right, so I've talked to you about how we can engineer the bugs that live in the gut. And so now let's move on to how we could potentially sense disease biomarkers, because we don't want this maybe producing it all the time. In order to understand this, we'll take a brief detour into yeast mating. So yeast can mate. It actually has two different mating types. Some yeast are type A and some are type alpha. Now, A and alpha both produce pheromones that can be sensed by the opposite mating type. When that pheromone is sensed, it initiates a program in that yeast to produce these protrusions called schmooze. Those will then eventually fuse and form a diploid yeast. Recombination can occur, and then further sporulation to produce haploids again. But in our case, we're very interested in how these yeast are able to sense those pheromones. Interestingly, the pheromone sensor in yeast is what's called a G protein coupled receptor. G protein coupled receptors are this very diverse class of proteins that can sense a wide variety of agonists. When they sense it, they release a G protein that initiates a transcriptional, a cascade that initiates transcription from a target gene. Now, these are very diverse. In fact, our own body has over 800 of them encoded in its own genome, and this is just the map of the ones we encode in our own body. So our idea was, let's use these G protein coupled receptors as sensors in this probiotic yeast. So we teamed up with a group in Denmark, led by Emil Jensen, who is one of the world's experts in how to tune heterologous GPCR expression in yeast. And so his platform basically involves taking human or heterologous GPCRs and fusing them to make a chimeric G-alpha protein. So you have to kind of have both the human and the yeast chimera in the right way, such that when you get the signal, it transduces along yeast's native mating pathway. What happens is once this heterologous GPCR is activated, you get transcription from the FUS1 promoter in yeast. And so you put GFP right after that. And if all goes well, you get a dose-dependent change in gene expression based on a new ligand. And so you've made your own inducible promoter in this case. Now, we had a problem, and that problem was Boulardii is diploid, but it cannot sporulate. It can't make spores that are haploid. And that caused a big problem because if you're a diploid strain, you are insensitive to any signal or any pheromone that you would like to put on it. So we had to figure out a way to get this to be a haploid cell or a single mating type without sporulation happening. It has a defective sporulation locus. Emil's great idea was to use Cas9 to disrupt the endogenous copy of alpha in its genome. So it was a diploid, but two type A's were present in both copies of its genome. So it was a type A, mating type A strain, but it was diploid, which is kind of weird. But the interesting thing that you can do with this strain is now it is sensitive to alpha factor, which normally would be only sensed by a haploid A-type yeast. As you can see, the output, which is a fluorescent signal, increases in proportion to the amount of alpha factor that we add. So what we were able to do at that point was say, okay, let's replace the native mating factor responsive GPCR with a GPCR from another organism. I'm showing three examples here. One is a mating factor GPCR from a related yeast called Schizosaccharomyces pombe that senses its mating factor called P factor. And as you add more P factor, you get higher gene expression in that case from Boulardii. Another two GPCRs that we decided to overexpress were an adenosine sensitive GPCR and a melatonin responsive GPCR. In both cases, we were able to see dose-dependent increases to fluorescence. Some of them work better than others. It's kind of an art process to get the right GPCR working the right way in any yeast, Boulardii included. But we think this is a great way to tune this yeast to sense diseases of interest. And so we're further studying this right now. Alrighty, we're moving on to chassis productivity. And in order to understand sort of what we were doing to improve yeast's ability to produce drugs, what I'm first going to talk about is our target for these drugs, and that was C. diff infections. This is a large group of collaborators, both at NC State and at UNC, who are dedicated to engineering this yeast to synthesize these antitoxin peptides. So I'll get into this in the next slide. This is just our picture we use for grant applications. Right. So C. diff infections, I think I'm preaching to the choir here, are a major healthcare issue. Some of the reasons why they're really difficult to eradicate from your body once you have it is that you have these really tough spores. These spores are intrinsically resistant to antibiotics. And so if you clear an infection with vegetative Clostridium difficile, the spores stick around and they can regerminate. These spores love to germinate in guts that have been recently been antibiotic treated, which cause them to be very common in healthcare settings. One of the things that causes disease in C. diff is the toxins that they secrete. Clostridium difficile just secretes toxins A, B, and also a binary toxin. And these toxins destroy your gut lining, leading to diarrhea and inflammation. And I don't have to say this again, but the relapse rate from the antibiotic treatment is one of the reasons that makes this such a big problem. If you have relapse once, you're more likely to get relapse again. So let's zoom in on these toxins that are causing the disease. These toxins are very large proteins. Most of this real estate, amino acid real estate, is devoted to getting this orange region of the toxin into our own cells. So this is secreted by C. diff, and the job of all of these other regions besides the orange region is to get the orange region into our own cells. So what does it do there? Once it's in our own cells, it glucosylates these key proteins in our own cells called Rho and RAC GTPases. Once these are glucosylated at a specific residue, they become inactivated. Once these are inactivated, the cytoskeleton of our epithelial cells breaks down. And once that breaks down, you now have movement of material from our gut into our body into our gut. And you can get infections, you can get sepsis, you can die from these types of toxins. And so our goal when we started this work was to find peptides that could inhibit the glucosylation activity of these toxins. So how are we going to do this? This was a collaboration with Dr. Menegatti in our department, and he has the ability to coat beads with peptides. You can make 10,000 beads at one time. Every bead is coated with hundreds and hundreds of copies of the same peptide, but every bead has a different peptide sequence. And so his idea was to color toxin A, so we start with toxin A, red, and if this toxin bound on its active site to this peptide, then that bead would turn red. But what we would also do at the same time is co-incubate these red toxins and colorless beads with a green substrate analog. This was UDP glucose fluorescein. So UDP glucose is the normal substrate of these toxins, and this is a green labeled UDP glucose. The idea being that if the peptide bound the toxin at somewhere other than its active site, the active site would be exposed for binding to UDP glucose. And so what we would hope to find are beads that are only red, but not red and green. So how are we going to find these beads? We turn to microfluidics to help us out here. We were able to generate these beads using a solid phase peptide synthesis, incubate them with our substrates of interest, and then what we did is we passed them through a custom microfluidic device that the Menegatti and the San Miguel lab at NC State had made. And so what happens is a single bead goes into an imaging chamber, and a computer decides whether to keep it or to discard it. Ethan Purnell helped us optimize the system for this purpose, and we were able to then sort through beads that were just red and not both red and green. So here's a representative picture of how we were able to do this. We had to actually tune the UDP glucose incubation period because UDP glucose is really easily diffusible. The toxin is much larger than UDP glucose, and so it doesn't diffuse very far into the bead, but UDP glucose diffuses readily into the bead. So we had to tune that a little bit. But what was exciting, this was work that was done in collaboration with Dr. Magnus at UNC, is that when we take these peptides and put them onto monolayers of primary human epithelial cells and measure resistance across that monolayer, what we see is that under normal circumstances, right, we add TCDA to this monolayer, you get a really drastic drop in electrical resistance, indicating that this monolayer has been disrupted. However, when you deliver one of our peptides, either LS5 or S1, it mitigates that toxicity, such that even S1 enables a much higher resistance than cells that had not been exposed to toxin at all. And so we were really excited about this. We published it, and we were really excited to continue this work and realize, wait a minute, TCDA is not the most clinically relevant toxin that C. diff makes. Actually, it's TCDB. And so we did the same thing. We did TCDA, we did it with TCDB instead. So the same group of people just fired up their machine again and did it with a different toxin. We found beads that worked really, really well, and we sequenced what the sequences were. Now, Carol Hall in our department is really good at computational optimization. She can simulate the activity of these peptides around the active site of these toxins and measure what she calls a gamma score, which is kind of like a binding free energy between the peptide and the toxin around the active site. And so she was able to take our hits we got from our experiments and make mutations to those sequences in silico to identify what we call peptide SB6. And this peptide had a very strong predicted binding to our toxin. And it binds, it's predicted to bind right on this sort of active loop of that toxin. That loop sort of sits right over the UDP glucose binding site of that toxin. And so we were really excited about this now. Okay, now we have the right peptide for the right toxin. Let's see in vitro how this peptide works. Carly is a wizard at enzyme kinetics and enzymology, and she constructed these beautiful Lineweaver-Burke plots showing the ability of this toxin to glucosylate RAC1 at different concentrations of UDP glucose, RAC1, and toxin. And so what she was able to find through these Lineweaver-Burke plots, if you remember your enzymology classes, is that this peptide was non-competitive with respect to UDP glucose. So the peptide does not inhibit the ability of UDP glucose to bind to the toxin. However, this peptide was competitive with respect to the substrate of that toxin, RAC1. So it directly inhibits, we think, the ability of RAC1 to bind to toxin. And so now what we're interested in doing is following up on this peptide to identify whether it actually works or not. So we turn to Scott again. Again, thank you, Scott, for doing all of these experiments with us. He was able to give this peptide to primary human colonic epithelial cells and show that, so these are control experiments here. We have cells that have been exposed to TCDB. You see they drop their tier at various time points. This is an overnight experiment, by the way, because you can notice what the time points are here. So it drops precipitously at T equals 13 hours. If you just give peptide without TCDB, the cells look basically normal in terms of their resistance. We can also give these cells toxin, but a peptide that shouldn't do anything, and it doesn't do anything. The cells still die. However, when we give our designed peptide, which is peptide 6 plus TCDB, it extends the lifetime of these cells in this model. So we have about, I would say, a 6-hour extension of resistance at that toxin B concentration. And the fluorescence images you show here are caspase staining. So if it's really green, it means that cells are undergoing apoptosis and dying. And so in this case with peptide 6, you get less caspase staining than you do for these controls. So what we're interested in now is sort of collaborating with yet another person who knows more about C. diff than I do, Casey Terrio, who hopefully can see if these work in a more relevant model of disease. So given that this was our target thing to produce, we need to engineer our yeast to produce it. Now, this is a peptide, and peptides go through a well-known pathway of protein translocation to get outside the cell. Now, we're sitting on the shoulders of giants here because yeast is one of the most well-studied protein secretors that's out there. Now, Dennis did an extensive literature search and sort of found every gene that has ever been modified in yeast to improve its secretory capacity. She made this list and she said, okay, I'm going to knock all of these out and see what their effect is on secretion. Now, this is no easy task because, again, this yeast is diploid, so she has to make 2 homozygous edits to this chromosome. She made all of these knockouts. They're shown here on the x-axis, and she decided to go fly to Denmark and test them in their microbioreactor setup that they have over in Denmark. So she worked with Jose Luis Martinez Reis on this, and she measured protein or peptide secretion in terms of nanograms per microliter. You can also think about it as milligrams per liter. So wild type has a relatively high secretion capacity, but a lot of these knockouts were able to improve secretion capacity to a large extent. In particular, the things we were really interested in were the last three of these knockouts here, YPS1, PRB1, and PEP4. They are proteases, so they have sort of a intuitive impact on protein secretion. Knocking out a protease makes it such that the thing you're trying to produce does not get degraded as soon as it's made. So we thought, okay, this is a rule we can exploit. Let's try combining these together and hopefully make a better secreting strain. This is the same data, just focused again on those three protease deficient strains. So with the help of Ibrahim in my group, we started stacking these edits in our yeast strain. So he made a PEP4 PRB1 double knockout, and that improved secretion. We were very excited about that. Unfortunately, when you combine them all together, they do not improve secretion. They're better than wild type, but they're not better than the double knockout. And so I said, okay, it's okay, Dennis, right? This is still a reasonable story. We can go home and write the paper. And she said, no, I want to try one more protease. This is a protease called APE1. We actually collaborated with Metric at NC State to do a supernatant proteomics study on our yeast. So we took all the supernatant from yeast and saw what proteins it was secreting. This is a protease that this yeast secretes that we didn't catch in our initial literature search because nobody had studied it before for this purpose. And so we said, okay, well, maybe we'll try knocking that out too. And lo and behold, when you have all four knockouts, we get this really synergistic increase to protein secretion. This is what we call the quadruple knockout yeast. And it makes about 5 grams per liter of peptide in our cell culture supernatants. We're further following up on this strain to see what other types of proteins it can secrete. So we're pretty excited about that. Ibrahim and Dennis, this would normally be enough for a thesis, but they wanted to do one more thing, which was to make inducible promoters in this yeast strain. And so the way that they were able to do this was to create these sort of synthetic yeast promoters that had a minimal TATA box, a transcription start site, and designer upstream activating sequences that would be responsive to the presence of small molecules. This is an example in hydrotetracycline and the TET repressor. When they express the TET repressor, it gets de-repressed by ATC, causing the production of a fluorescent molecule. Now they did this for several different small molecules. Actually, CUP1 and GAL1 are native to yeast. So those didn't need to be engineered, but lactose, xylose, and hydrotetracycline were sort of these designer ones. And so they did this orthogonality test where they tested not only the inducer that was supposed to activate the promoter, but other inducers that were not supposed to activate that promoter. And we saw in several cases we can choose pairs of these that work really well together. So Dennis, what then she did is he took one of these that worked really well as a galactose inducible promoter, and she put it into mice. These mice were antibiotic treated, so they were not germ free, but they had some bacteria inside them. And they gave this either wild type yeast or yeast that expresses nanoluciferase under the response of the galactose promoter. These were either uninduced or induced with galactose. And we took one of these to the imaging core at UNC. Again, thank you, UNC, for helping us out with all of this. And we did a sort of a bioluminescent study on this nanoluciferase. So SB-Gal plus by itself, this is a yeast strain that does not express nanoluciferase, has sort of that basal level of a nanoluciferase expression, has sort of a basal level. And then the engineered strain that is uninduced kind of activates a little bit in the gut. We think this is due to residual galactose that might be present from the mucus, and I'll get back into mucus a little bit later. But finally, when we give these mice galactose, they substantially induce their gene expression. You see also, we think these are where yeast is more localizing in the gut. You see these sort of pockets of where yeast likes to be in the small intestines and also in the cecum. So we're pretty excited about exploring the biogeography of this yeast a little bit further from these studies. But if you just want sort of a quantitative aspect to this, our findings were that when you add the inducer, you get substantial increases to expression versus the uninduced condition as well. All right, we're on the home home stretch. Thanks for sticking with me through all this. This is a rather short section, but it's something that relates to the previous section about colonization. So colonization I'm sure you all know, if you're a microbe, colonizing the gut is not an easy task, right? You're made in an environment that is very nutrient rich, non-competitive, but then you are transported into an environment that has quite the opposite. It has many competitors. Its nutrient source is heterogeneous. There's flow that's sort of pushing you out at all times, and there's an immune system that you have to deal with too. And so our conundrum for us is what of these features is most limiting Boulard's colonization. Um, Mucus is one of the things we latched onto as a potential solution to this problem. Now, mucus is composed of these highly glycosylated protein chains. They're made from primarily 5 different sugars, but they're all arranged in different ways. Now, mucus is an important food source for many microbes in our gut, and unfortunately, Boulardi can't eat any of them. Actually, it looks like we tested them on all 5 of them. Galnac looks to be a positive, but we've confirmed this, it's not actually a positive. So this yeast can't consume any of these mucus sugars. So our goal was to get this yeast to eat at least one of them in the hopes that it improves its ability to colonize the gut. So of these 5, we chose galactose to start out with because Boulardi, unlike baker's yeast, is unable to eat galactose. Now, baker's yeast can eat galactose just fine, but Boulardi is interesting because it has a defective PGM2 gene, which is part of its galactose metabolic pathway. When PGM2 is mutated in this way, it has a premature stop codon, this yeast cannot grow on galactose, which explains the very bad growth curve you see on this picture. So what we did is we corrected that premature stop codon, and we tested its ability to grow in galactose. Now we're comparing in blue, wild-type Boulardi, versus green, which is SB Gal+. This is our galactose-consuming Boulardi strain. And in both 30 and 37 degrees C, we see that SB Gal+ can grow on galactose very well, whereas SB cannot. And it can grow on both sugars. So you see this sort of characteristic dioxic shift here where if you add glucose and galactose, both strains can grow until the glucose is exhausted. And then our SB Gal+ kicks in to consume the rest of the galactose. So we wanted to know if this could colonize any better than our wild type strain. And we were very interested to see, we had 4 groups of mice, 2 that were antibiotic treated either with wild type or the SB Gal+ strain, and then 2 that we had added galactose to either the wild type or the SB Gal+ strain. And so to make a long story short, what you see is that when you add galactose to wild type SB, it actually inhibits its ability to colonize the mouse gut. The purple line is very sort of low in this case. Whereas when you give galactose to the SB Gal+ strain, it actually increases its ability to colonize versus the wild type strain. So that was an interesting sort of modulator of colonization here. One thing that we saw that was quite interesting was that if you add, if you add galactose to SB Gal+ and look at the small intestine, you get much higher small intestinal colonization levels than we've ever seen before for this probiotic yeast. So small intestine is a important source for nutrient absorption, and so we're thinking that this idea of engineered mucus catabolism could be useful to expand the biogeography of this yeast. Whereas with the strain that is unable to consume galactose, but you give it galactose, it actually really reduces its ability to colonize the gut. And so to wrap this all together, we think that beyond just galactose, perhaps these other mucus sugars could be useful as well. So I've walked you through all 4 of these aspects that are sort of the nuts and bolts of how to get at this dream of using the gut as a drug delivery platform. We're certainly not there yet. We think we have a long way to go, but we're having fun while we're doing it. So I want to thank you all for attending the seminar. This is a picture of our group on last Halloween. So we like to do things, we like to have fun too. And I want to thank in particular the people who helped us with this work. Dennis, Ibrahim, and Scott were the students that were involved. Carly's near graduation also helped with this work as well. I want to thank all of our funding sources for taking a really big risk on us, and hopefully the risk was not in vain. And I want to thank you for your attention. I'll take any questions that you have. So thank you very much.
It was a great talk. Thank you. Uh, for folks who might have questions, feel free to unmute yourself and ask or put them in the chat, and I can take the questions. But in the meantime, let me ask you a first question. This is really beautiful work. I'm really curious. I mean, I'm sure you're working on this already, but I'm wondering how engineering yeasts to use substrates like galactose, which are nutrients for carb for other bacteria in the gut. How do you think that would affect the functional of the indigenous microbiome?
Yeah, I think it's a question we're concerned about. Where we haven't been able to quite yet engineer the yeast to consume anything other than galactose. We're working on ClickNAC right now, but the thing we're really interested to study, like you mentioned, would be to look at this 16S abundance of the bacteria in the gut. We are encouraged by the fact that commensal candida can consume some of these mucus sugars. We're hoping to maybe utilize its niche space. Maybe I'm okay booting out commensal candida in favor of Boulardii. But that's sort of a question that we always have to sort of test its safety once we have those strains. So it's a good point. We don't want to be messing up the gut too much by utilizing all those nutrients or degrading the mucus layer for that matter.
Yeah, Scott, go ahead.
Yeah, that was really great work. I was wondering, in the experiment that you did in the notobatic mice, did you see any sort of inflammatory response from just the native yeast because they were, there were so many colonizing the gut at that time?
Yeah, that's a great question. We did not do we didn't look there, and I wish we had. We didn't see any sort of gross, I would say, behavioral changes in those mice. They looked healthy. I think I remember asking my students like several times, "Do they look okay? Do they look okay?" They looked okay, which was surprising to us because they were all yeast in their gut. But I do think that it would be well warranted to check in finer detail what the impacts on the intestine were in those types of experiments.
Do you know if any people that have immunocompromised conditions get overgrowths of any of these?
Oh gosh, yes. If you're immunocompromised, you should not take this bug. There are case reports out there of Cerevisiae fungemia, Boulardii fungemia.
Candida, big one.
Yeah, Candida. So it's something where you want to be very, very careful if you're immunocompromised. These sort of things are, tantalizing prospects, like the genes involved in, for example, invasion that might be turned on in a fungemia type model. You could potentially imagine using an engineering to do that, but we're not really anywhere near solving that problem with this. So if you're immunocompromised, I would definitely not recommend.
Ian, you had a question?
Yeah, very, very interesting stuff. So I remember you were showing some graphs in the beginning of the talk about the colonization of your mutant versus the wild type fungi. And it seemed the colonization was relatively regular, but I didn't notice there was a kind of a spike in the production of your peptide, right? In the early days that didn't seem to follow the trend in colonization. You didn't seem like you were losing the bug at the same rate that you were losing the production of your peptide. And I was wondering if you think that was there something I was missing there? Are you losing the bug in your colonization during that period, or is it reducing the amount of peptide it's producing in that niche for some unknown reason?
That is a great question. We don't know the full answer to this. I can give you some auxiliary information, and that will hopefully scratch your itch a little bit. But so this is sort of the vitamin production. Now that, yeah, you're right, the yeast is still there, but it's producing less of that vitamin over time. Now, things that we did that were a little bit that could have impacted this, where we were producing the vitamin from a plasmid. And it was a plasmid that is maintained using a uracil oxytrophic marker, which means that the Boulardii itself can't make uracil, but the plasmid enables it to produce uracil. Our thinking was that we would not see long-term production of this beta carotene because uracil should be present in the gut, and the yeast wouldn't need that plasmid to produce it. Nevertheless, even without selection, it appears that the plasmid stuck around. We plated these feces at the last time point, and all the yeast we were able to recover from the species were orange. So it didn't appear that they had, I would say, removed that plasmid. That being said, for some reason, this production does spike. Variance is relatively high here as well, but we don't know why. I wish I had a better answer for you.
Is that just related to beta carotene stability and like it's like turnover or?
Oh, yeah, we had issues, again, the gut probably had an impact on the beta carotene's lifetime. We also had issues with sort of storing our samples in a way that maintained beta carotene. So we had to store them all in the dark on dry ice, and it was a very big process. So that could have been from the storage as well, like you said.
I guess another more theoretical question, as a non-microbiologist that often finds himself in rooms with a lot of microbiologists, I hear a lot of conflicting ideas about horizontal gene transfer in fungi and potential of gene transfer even between eukaryotes and prokaryotes. And is that something you believe in or worry about in these situations?
That was a question too.
I believe the reports that it happens. I do not know the rate at which it happens. Right. So between fungi, I totally agree with, right? They mate. Between fungi and other types of prokaryotes, I believe that it's happened. You can see that in the literature. I don't know the rate, right? So the rate is what matters here. The other thing that as sort of another point that might further complicate this matter is that this yeast doesn't sporulate. We don't think it mates, right? You can't get this thing to mate with normal yeast under normal circumstances. And so it's potentially a little bit safer in that regard. But when this yeast dies, the cell wall will start breaking up and potentially also having some ability of other bugs to take it in. I do think it's an important thing to get quantitative kinetic data on, because that's the only way you actually do useful things, if you know how fast it goes.
Can I go?
Yeah, go ahead.
Thank you. Hi, Nathan, Sue.
Okay.
A comment and a question. So since you have this beautiful slide about the vitamin A production up, I'm always concerned with things like these experiments with off-target effects. The vitamin A metabolites are, I think, now accepted to be the immunoglobulin isotype switch factor for IgA. So I wish we had evaluated IgA levels in these mice. It would have been really interesting. Could be beneficial, might be detrimental, who knows? And Scott, I was there when Dennis euthanized these mice, and I don't remember any adverse macroscopic effects, which is interesting now, I realize. My question is, and this might be a really naive question, I might have missed the answer, but when you did the quadruple knockouts, are those yeast going to secrete much more of all of the proteins that they make? And again, is that going to be an issue for off-target effects, or can you engineer them somehow to just make what you want?
Yeah, that's a great question. We were doing this via ELISA and measuring production via ELISA. So it was hyper specific to the thing we were interested in. I have a student now, Will Parker, I told him, I want to see a gel. Show me a page gel of a supernatant. And so if what you're saying is correct, and that's just secreting everything, then you should start to see a much more messy looking page gel. And so I definitely am interested in that. If in terms of getting it to secrete just what you want, I don't have any good ideas on that at the moment, but it might be a trade-off between secreting boatloads of everything and nothing, right? So maybe there's a sweet spot with those knockouts that we found. where it secretes much more of what we want, but not as much of the as the other stuff. So it's a great point.
Good. Thanks.
There's a question in the chat. Can you expand on why, and this is from Jalu, can you expand on why your engineered yeast is able to colonize the small intestine more? I wonder if it has anything to do with the redox and with the small intestine being a more oxidized niche.
That's a great question. We have routinely not observed under normal circumstances, this yeast doesn't have a high load in the small intestine. Our thinking was it wasn't able to colonize there because the transit time of the small intestine was much more rapid compared with the large intestine or the colon. But that was just a hypothesis. When we see the increased abundance in the small intestine, it's because it's in the mutant that was able to eat the galactose. And so you're probably right in that if there's more oxygen around, the yeast can grow faster. I mean, that's a thing that yeast does. It likes oxygen. We're thinking also that the ability to eat the nutrient sources there can play a role too. But that's something we again have to follow up on to figure out why.
Mm-hmm. I have one more question for you, Nathan. Could you just briefly talk about how glycosylation differs in yeasts versus mammals? Just, you know, and then secondly, do yeast GPCRs have the same type of inhibitory and activating subunits for their GPCRs that, you know, higher like mammals do?
Right. I'll tackle the first one first, and it impacts how relevant the secreted products are. Right. So when you produce a therapeutic in mammalian cells, it has a human-like glycosylation profile. Now, yeasts love to their glycosylation profile is not like human cells. It actually produces much more mannose heavy sequences on its proteins. So if the activity of your protein is dependent upon the glycosylation status, then you probably want to be very careful with using a yeast-based production platform. I can say glycosylation matters a lot for protein immunogenicity and lifetime, particularly in the blood. I'm hoping that the environment of the gut is such that it's a bit more tolerant to these sort of alternate sugar forms. Activity aside, hopefully the immune system is able to tolerate things that have mannose on the outside.
Yeah.
Related to the activating and deactivating GPCRs, yeast only really has two GPCRs. One is a glucose responsive one is a pheromone responsive one. I don't know if those have the same activating or deactivating sequences, and that's really part of the challenge with expressing these human proteins is that they're much more complex than the yeast ones are. You kind of have to truncate bits off that you think are not relevant and hope for the best from the thing. So it's not a high throughput operation by any means.
And the reason I was asking about that latter question is because, you know, can this be analogous to what's done in neuroscience, like making dreads, you know, designer ligands and dread receptors?
Oh,
you know? So yeah, I wonder if your system is amenable to making designer GPCRs with, you know, bespoke ligands. I mean, you kind of alluded to that already, so you know, clearly it's possible, but yeah.
Yeah, here we're trying to choose our battles. You're right in that people have been thinking about this with yeast, but there are people who are like in the Jensen lab and in other labs who are thinking about doing directed evolution on these and getting them.
Yeah. Great. Very great. Amazing work. This is so exciting. Is there anybody else who has any questions? I don't see any coming up, but thank you so much for presenting to the CGIVD community. We really appreciate it. And yeah, see you around.
See you around. Thank you very much.
Yeah, everybody take care and stay warm.
Yeah, see you soon.